give the first word to Francisco Moyano, who is the father of in vitro digestion in fish, and he has been, and so he's the person who knows more about it, and so he can tell us what we've done correctly, what we've done wrong, and how we should progress, and what else we should look at in order to improve this part of the project. Please, Francisco, thank you very much.
First of all, I would like to thank Fulvio and the rest of organizers of this very interesting workshop to have invited me to be a part of this revolution in the in vitro focus on aquaculture. To be honest, I feel a little bit old in this sense. I feel the risk of seem a little bit pretentious. I feel like a pioneer in this because we have been, me and my group, we are working with in vitro trials for 20 years. And we felt a little bit alone because nobody was very interested, and this includes also the editors of the journals, but they were not very interested in the in vitro subjects and the in vitro trials. But now I'm very happy to see that a lot of people, a lot of very good researchers are putting efforts and refreshing this issue. And I'm sure that in the next years, we will assist to a revolution and a greater use of those techniques. And well, here I see that what I told you yesterday, Fulvio, that a strange coincidence between our logos. But mine one is older. Well, only 5 slides to say general ideas that some of them were also exposed this morning. So our part of the in vitro trials was focused in the hydrolysis part, nor in the absorption part. So this is important and has to do with all the results that Amir and his group presented us this morning. So we say that we follow the conventional trend in nutrition in human and terrestrial animals. We decided to follow the lines that were used for pigs and poultry and also for humans. So plus pay more attention to the chemical transformation produced by the digestive enzymes and by the environment that is present in the gut to the absorption process. And the parameters that we focus to evaluate are the bioaccessibility and the potential bioavailability. So here we have the more or less a definition of what we understand as bioaccessibility of nutrients. It's the fraction of a given substrate that may be accessed by the proteases, amylases, and lipases, the digestive enzyme, and also to make an estimation of the potential bioavailability of compounds. So the products that are released after hydrolysis may be potentially absorbed by the intestinal epithelium. So this is the reason why our assays can be connected and must be connected to the platforms that are mostly specialized in testing the absorption processes. Our platform has also a great coincidence with some of the designs that were presented this morning by Pedro. They have also several years of running. Also are chambers that are bioreactors formed by 2 chambers, one hydrolysis chamber and one down chamber that recovers the products of the hydrolysis and is separated by a semi-permeable membrane. And well, here we have the arrangement, and we have also, it's not present in here, also we have the pumps to establish a flow of, in our case, is a buffer solution that is continually draining and recovering the products of the hydrolysis. Okay, so those are this is the hardware, the semi-permeable membrane small bioreactor, and also we consider the hardware, the biological material that we use, so the enzyme extracts. In our case, we have focused a lot in optimizing this part of the assays because we are not focused in only testing one species, as in the case of the platform in the rainbow trout. We worked with a lot of different species of warm water species, freshwater, marine water species, even shrimp, octopuses. So we need to develop a protocol to prepare the enzyme extracts to be used in the in vitro assays. So this is what we call the software of our platform. And so where we are focused on and establish good protocols to separate, to collect the enzymes at the more suitable moments and to prepare the enzymes and to characterize their activity to be sure that they can be used repeatedly and with accuracy. So for this reason, enzymes are collected at suitable moments. So it's very important the moment on which you collect the enzymes when you have a fish, because the digestion is a dynamic process. So depending on the moment after the meal intake you collect the enzymes, you can collect a completely different profile of, let's say, stomach enzyme or intestinal enzymes. And the amount also is important, because depending on the moment, you can have a peak or you can have a valley. So if you want after to correlate this and to prepare, okay, let's simulate a 100-gram fish, you need to know which is the average production of enzyme per gram of fish you have in this species under normal conditions. So normal feeding conditions, and you need to take all those samples before in a very established and closed manner. So this allows us to estimate the enzyme estimation of the total production of the enzyme that is corresponding to a given intake, because this is another important point, the enzyme substrate ratio. Not any amount of enzyme can be put in the bioreactor. Okay, you can put any amount of enzyme, but this is not representative of the real situation in vivo. So if I'm going to simulate a 100 grams fish, I need to put the enzymes corresponding to a 100 grams fish, and also the substrate that should be ingested by this size of fish in one meal. So those should be the physiological enzyme substrate ratio that can produce a digester that could be representative of the real situation, okay? So the physiological pH values are also important. Another question that is very important, and some of the assistants know this very well, the variations in the pH along the digestion are very, very big. We can use any pH. Okay, let's put an acid pH for simulate the stomach. Let's use an alkaline pH to simulate the intestine. But this is not real. For example, in the case of sea bass or sea bream, for most of the stomach digestion, the pH may be around 5. This is not the optimum pH for the action of the pepsin. So, but if we are going to simulate this, again, we need to take this into account to get as much closer as possible to the real situation that we have in vivo. The other point should be the ionic environment. So we need to follow the steps that the researchers in terrestrial animals and also in humans have developed. They have focused on, they have been pay a lot of attention in providing a suitable ionic environment using salts. Let's say the concentration of ions, let's say sodium, potassium, calcium, even other minor ions that can be useful and needed for the optimal performance of the digestive enzymes, and also at a concentration that mimics that existing in the gut. Also, and the last of the points should be how to mimic, how to consider the real hydrolysis time. So this must be focused also, this must be based also in the real gut transit rates, or the real time that lasts the digestion, the complete digestion process in a given species. Of course, this may take 1 day in a real situation, but we need to at least to know which is the length, the relative length of the stomach stage compared to the intestinal stage, and to consider this to compress this, and okay, let's run the assay for, let's say, 10 hours or 8 hours, but consider that the first third part should be the stomach digestion and the rest should be the intestinal digestion, because if not, this should be, again, not representative of what occurs in vivo. Well, yeah, we have also our expertise, more than, as I told you before, more than 20 years working with those systems. with the very initial configurations to a more sophisticated platform that we work today. We have published 23 papers, book chapters, contracts with companies. This is a very important thing because this demonstrates that some companies may be interested in using those assays for testing mostly bioactive compounds or some special additives that they are not, it's not easy for them to develop the in vivo trial. So this is a good potential also for the platforms based on the use of cell cultures. And we have developed a lot of different assays, nutrients bioavailability, mostly in protein. We have been working also in the ranking. We have been talking this morning, ranking for protein ingredients. We have been working with a company for 3 years, for example, that they wanted to have a standard methodology to compare the potential nutritional value of the protein fraction that they buy when they have different feed providers. So they wanted to compare this using a standard methodology. This helped us to improve our methodology to include internal controls to be sure that we were repetitive in our assays. And also, in the case of those assays that we are asked for by companies producing bioactives on other compounds, we were mostly focused in the testing of the effect of digestion on antioxidant compounds, like phenolic compounds, also any other mostly phenolics, and also those bioactives coming from plant ingredients that may be also very reactive with proteins and other compounds that may be present in the diet. So some of those ideas were exposed this morning by different of the presentations. So I only want to reinforce or to highlight the need for any platform that is aimed to simulate in vitro the digestion of the fish, to be flexible, to be simple and adaptable to a variety of operative conditions. In our case, this means that we need to move and to change, as I said before, from one species to another. And also to change, for example, asking, okay, focus only on the stomach digestion, because I need only to see what happens after the acid digestion. Focus only on the intestinal digestion, represent all the whole digestion. So you must be also flexible in this sense. Replicability, of course, we need to be able to repeat our results under the same conditions. The accuracy, and this I want to make to highlight this in this case the relevance of testing not on ingredients alone, but within the matrix, because we have realized that the things may change a lot if you test, let's say, for example, an ingredient alone, or this same ingredient within the normal matrix in combination with the rest of ingredients that includes lipids, carbohydrates, the famous cage effect produced by carbohydrates. Also, the interaction of those ingredients with the conditions of the preparation of the feed, that means heat and that means pressure. So, and the release and then the potential bioavailability of compounds may be greatly modified. So we need to consider this. And of course, we will talk about this during the season. The need for the standardization. But this is the same requirement that we have in vivo. We need to standardize all the conditions. If we need to the reliability and of the in vivo digestibility trials needs also that we need to use exactly the same conditions and to compare the results. If not, it's not possible. And well, some ideas for the future should be more or less the same. In vitro assays will be increasingly used. I'm sure from this meeting onwards that this will be true. So the platforms may be adapted or must be adapted to simulate the digestive feature of the main commercial species. So there's an open window not only to apply for trout or for salmonids, but also to other species, for the Mediterranean species also, and also for warm water species like tilapia, because there is a great potential to use this in a big market that may be the Asian or South American fish species. And Platforms must combine hydrolysis and absorption stage, so the models that we have been talking this morning with the hydrolysis model. And we need to arrive to a consensual methodology that may be or even formed not for one type of platform, maybe different types. This morning, we were some results were presented that using different cell lines, you can obtain different results that may be more adapted to the nutritional evaluation or the health evaluation of some products. So this could be a good example of this. And those should be a sort of portfolio to offer to the companies that may be interested. And this should be all on my part.
Thank you very much, Francisco, for your very comprehensive introduction. And now the discussion is open. You raised a lot of points. Our question from the public. Do you have questions to the members of the consortia or aspects that we should have explained more in detail or things that you want to know more in detail?
No, yes. I asked them, and we were talking yesterday and also during this morning, the constraints using times. And also, for example, the inactivation of the times that they are going to be used following the next step that we did in the cell culture, because I think that there may be a sort of interaction between the components of the digestion. No. Do we repeat or it's okay? No. This morning I have a question about the how the inactivation of the digester that you obtain can release or produce some harmful products that could interact after with the medium that is used to feed or to maintain the cell culture. Because at the end, you feed or you use a bath of nutritional liquid for the cells that contains some nutritional compounds for the cell mixed with the digestive. So this interaction may be quite strange. How do you assess this point?
Thank you. Thank you again for your help at the beginning of the project. You're right. I mean, when we knew that we need to activate the enzymatic activity in order to protect the cells, we have two options, mainly. One was the heat inactivation or using this PFA block, which is a protease inhibitor. The problem with the second one is that we will inhibit also the activity of the enzymes in the cells. So we opt for the heat inactivation, and it's true. We don't know exactly what is going on in the system. You saw that we have increased toxicity when you increase the concentration of the digester, but we don't know exactly what is causing the toxicity. It can be toxic elements produced, as you said, by the heat inactivation. It can be also the osmolarity that we know we have measured before, and it's very high when you increase the digester concentration. And also we cannot rule out the presence of other elements that can be toxic for the cells. So it's an open question.
May I add a little to that based on some questions from the online attendees? Because I think, and which also relates to the digestive process of this, and a question that I think has been brought up in the Fish AI consortium all through these 5 years. How close to physiology do we need to be in a model system like this? And for example, we have questions online. For example, how close do we need to adjust the pH to reflect normal physiology of these AI models?
I think as much as possible, because this really affects the action of the enzymes. For example, in the case of stomach enzymes, you can establish, okay, the optimal pH for pepsin may be 2 or 2.5, but this is not the real situation in the stomach of a zebrin. So if you are trying to simulate, to mimic what is occurring in the stomach of a zebrin, you cannot put a pH of 2, because in the real fish, most of the protein is not hydrolyzed in the stomach, but in the intestine. So it's only a small, a very slight predigestion of the protein. This is completely different in the case of salmonid fish, on which a low pH can be reached in the stomach, and the pepsin is very active. So the protein hydrolysis is completely different. This is what I know.
Okay, so pH, yes. I'll follow up with another question here. What about temperature? Do we need to consider temperature, or the question is actually, can we run the in vitro systems under, what to say, the normal physiological temperature of the species we are studying?
It's a good question, because we need to find a compromise between maintaining the real temperature, also using the same schedule. Okay, let's run the assays in rainbow trout at 15 degrees and also in tilapia at 30 degrees. But this means that an assay for trout should take 1 day or 2 days. So in our case, from a practical point of view, we try to develop in salmonids at 20 degrees only to increase the activity of the enzymes, to get a response in a shorter time. But in the case of warm water fish, we try to maintain the real water temperature. For example, in the case of tilapia, 28 degrees. In the case of sea bass or sea bream, 22, 23 degrees. That is a normal temperature for growth of those species.
Francisco, what about salinity? Because we know that salinity has an impact on digestibility, because even the amount of water a fish can drink can affect digestibility. So in this kind of models, do you take these into consideration?
Yes, for this reason, I talk about the importance of the ionic environment. So we include, for example, in the simulated juice we use to develop the reactions, we add the enzymes, we add the ingredient, we use a buffer solution for the intestinal stage that is includes also sodium, includes also calcium, and includes also taurocholic acid, also a bile salt that is also interesting to simulate what are the conditions. And we even have developed some in vitro assays simulating the effect of increasing concentrations of sodium chloride. For example, it was done for Lisa ramada, a sort of mullet, species of mullet. And we realized that it was affected by the salinity. But this is the good point, because we are able to simulate the condition of different intakes of types of water intake. So for urealins, stenolins, so more salted. So the good thing is, okay, you can decide which kind of ionic environment you want to simulate, and you will obtain quite different results in terms of the efficiency of the enzymes. So you are right in this point, but the good thing is that we are able to simulate this in a more comfortable manner than using a live fish.
On campus audience, please raise your hands, because we have active people here online, so I can follow up with another question that relates to this. So there is a question here also online which relates to the in vitro digestion, I guess. How to assess the buffer capacity of the pellets in vitro?
We consider this also because in the preparation of the assay, we try to make an initial estimation of the buffering capacity, because depending on the ingredients, this means that you need a higher or a lower amount of hydrochloric acid to reach the desired pH. So in the preconditioning of the sample, prior to add the first set of enzymes, let's say, for example, the stomach enzymes, we decrease the pH to the desired pH, let's say maybe 4.5, using the different amount of hydrochloric acid to reach the same pH. But this is a stage that we take into account, because, as you say, the buffering capacity of ingredients may vary a lot. So we consider this, of course.
Can I take advantage of Francisco's experience and raise the issue of origin of enzymes? Amir briefly mentioned this morning with this metabolomics that depending on the origin of enzyme, you have very different results, which include using exogenous enzymes, so not fish enzymes, or if we want to be more specific, we use rainbow trout enzyme, but in one cases we got them for very big fish, in some other cases in smaller fishes. And since we want to keep on going, one of the issues is that should we sacrifice fish just for getting the enzyme out, or should we use available sources like at slaughterhouses, in which case we have no control of our sources. So would that be too limiting? So it's better to get a ad hoc sampling rather than a random sampling.
No, it's a must. You cannot. I don't advise you to use slaughtered fish because they are starved for several days, so the amount and the quality of the enzymes may be very bad. So we say that we have fish donors, and we maintain a population of fish specifically as donors of enzyme, and we maintain them in suitable condition. We feed them for several days, and we sample. The first thing we do is to know more about the dynamics of the production of the enzymes for this size of fish. For example, in the case, some of the attendants make this kind of assay as a preliminary step to know, okay, I give a meal to a group of fish, I make a sequential sacrifice and get information about the variations in the pH and the variations in the production in the stomach and in the intestine. And now I know for the next donors, which is the more suitable moment to get the best sample for the stomach and the best sample for in the intestine. And knowing which is the amount of enzyme produced by the fish, I can have an information about total production of enzyme expressed in units per gram of live fish. And this is what we have as indicator for the next assays. This is one of the standardizations we do. But we need to run this using specific fish that are very well managed and sampled, and we know which is the most suitable moment to get the maximum of the fish. If you use any kind of fish, if you don't know the origin of the fish and the feeding status of the fish, you may be sure that you will obtain enzymes that may be very different from one pool to another.
So Francisco, considering that you need to have your pool of donors at different environmental conditions, like salinity and temperatures, because probably depending on where they live, they'll produce different concentration of enzymes in order to ensure that they have enough energy. So it's challenging.
This should be the ideal situation, but the real situation is you need to have some friends that are able to maintain some tanks and to maintain your donors, and you say, okay, you have 3 tanks. Do what you want. Okay, it's enough with 3 tanks. So the ideal situation should be this. But this means that in the case of you don't have the possibility of doing this, you can obtain this information along several years with different pools of the same species. Let's say if we have this for sea bream of this size at this moment, but if I have the possibility, and I did it for one species that is very common there, sea bream, because we have several batches of sea bream with different sizes, maintained at the different, and we obtain the enzymes of them and we characterize them. So we have a sort of base data of the different sizes and production, and we have a fork. Okay, this is the more or less the average obtained under different conditions. So the ideal situation should be, okay, to have this information that is basic to be more accurate, to get a higher accuracy when you develop your model. This is true, but it's a compromise between the availability and the desirability.
I believe we have a question on the top row.
Yes. I have a question about the buffer capacity of the ingredient. High capacity is better or low? Because in low buffering capacity ingredients, they adjust to much sooner than the other to pH level, for example, in intestine part or stomach. But I didn't get that. Is it better to have high capacity or low capacity? Which one?
It depends on the pieces, but in general terms, the lower the buffering capacity, the lower need to produce hydrochloric acid to decrease the pH to the functional pH, for example, the pepsin that must be below 4. So in terms of physiological response, the lower the buffering capacity, the better for the efficiency of the stomach digestion. It has not much impact in the intestinal stage, but this may have an impact in the live fish if the fish relies a lot on the stomach digestion. This may be the case for salmonids, but not in the case of tilapia, for example, or for mallets, on which the intestinal digestion is the more important part. So the stomach digestion is not very important. So the buffering capacity of the diet is not so important in the same way.
Yeah, perhaps I have another question online that aligns with this. It was actually a question raised to Ramon this morning, but I think it belongs here, and it's more to the industrial applications. So the question, I guess, to Ramon to answer is instead of an absolute in vitro digestibility value, for example, if you're testing a new fish meal batch, would it be enough to check by in vitro methods if a new batch matches a reference batch assessed previously in vivo?
Well, I don't know about about your in vitro methods. This I cannot talk about, but I know about our in vitro method, which is NIR. Yes, actually, we know the digestibility of our fish meal by NIR. We don't need to run in vivo trials anymore. We do trials to maintain the calibration, but our calibration is so accurate that we know exactly with a very good precision, we believe, the protein digestibility of fish meal batches, yes, and amino acids. Yeah. So we don't need to run in vivo trials. I mean, we do it for calibration maintenance, but not on a regular basis.
Can I add something to this question? What about complete feeds?
I'm not, well, we are very much focused on raw materials. And as I explained in my presentation, we assume that the digestibility of the different raw materials is additive. And by knowing the digestibility of the raw materials, we can estimate the digestibility of the feed. That's how we do it.
Are there any other questions from there? Otherwise, I can open up a chapter within this discussion, so which is related to this session, but also more widely. We have worked on rainbow trout, good model for salmons, but then there are a huge variety of other different species. And from our experience, it's probably quite complicated to get cell line, suitable cell line for every species of interest. What about the compromise of using available cell lines, but with species-specific enzyme? Is that a possible compromise? Would that give interesting information? At the end of the day, at the level of the single enterocyte, would there be lots of difference between a trout and a sea bass, for instance? Or at the cellular level, they work more or less in the same way? Whereas the difference is what happens in the lumen, so at the enzymatic level.
In my opinion, the closer the species, the better. So it's better than using caco cells, for example, than humans. So I think that it could be a good compromise solution to use the hydrolysis part adapted to using the enzymes of the species, and for the second part, the absorption for fish intestinal cells that even being very different or quite different, may be closer than using any other kind of mammal enzymes. It's the same question that using mammal enzymes to simulate hydrolysis, simulating fish species. I have seen papers, I have read papers published that the in vitro simulation of the rainbow trout, okay, they use sigma enzymes produced with pig. It has no sense. So this is crazy. But in this case, it should be a good compromise and an initial step during the development of perhaps a methodology that can produce suitable species and species-specific enzymes. But it could be a good compromise, in my opinion.
It's a compromise, in fact, but when we look at morphology of the intestine of different species, it's completely different. So when we are talking about absorption, you have different absorption rates depending on the integrity of the cells or the type of cells. And in fact, in many species, we don't even know what kind of cells they contain. I mainly work with marine species like sea bream and bass, and we try to characterize them. And it's not very easy because in other species you have markers for everything. You go to European sea bass, for example, we are trying to characterize the lymphocytes, and you don't know what kind of cells are there because they don't mark with, say, markers that are used for other cells. So the lack of knowledge is huge. We are in very early stages of understanding the morphology, even in vivo. So to arrive to an in vitro model that can simulate what we still don't understand well in vivo, so it's still a lot of things to do, very, very difficult.
Perhaps I can follow up a little bit with another question from online attendees with regards to what we don't and what we do know and what we don't know, and also how this applies in vitro. So you have seen this fish AI prototype. That is one example of an in vitro system. So the question, I guess, to us been working with that is, what happens to the produced metabolites in the in vitro model? And is this something that should be balanced, is the question. So I guess the question relates to when these cells in vitro are exposed to in vitro digested material. Of course, we don't take it away. What happens with it? Did we characterize it? Does it affect the cells and the functions and the downstream analysis with their interpretation? Would someone like to answer?
Well, I can start and maybe somebody will follow. First of all, definitely we might interact. The other thing that we hadn't got time to show is that the average time in which cells are exposed to catabolite are 3 days, because we change the medium every 3 days. So they will be renewed, and we also change the in vitro digest state. So we start from scratch every 3 days, or less if we shorten the time of exposure. So waiting for more complex platform that might include circulating aspects, which would, on the other end, bring a lot of other complications. So it's always a compromise between the ease of use and the definition of the problem. I guess that, as we mentioned before this morning, playing around between concentration and time of exposure will give you the optimal compromise. In this case, we've seen that at the high enough concentration, a 24-hour exposure is very informative. And we also seen that looking at things every 24 hours is very informative. So at present, we are moving towards that. So that kind of problem will be very, very limited, because we'll only be 24-hour exposure, and then we change everything. But other comments are very welcome. Amir agrees with that. Anything else? Okay. So Nicole, do you want to add something after you have done this many, many times? As they all agree, that's nice.